I'm Barbara Mason. I am a professor at the Scripps Research Institute in La Jolla, California.
So, how have you moved along your path in alcohol research? You can go back as far as you want. Adolescence, maybe high school. Were there people who had a special impact on your decision to become a scientist, to go down this path?
Well, when I was in graduate school, I was assigned for a year to train at the Smithers Center for Alcoholism Treatment at the Roosevelt Hospital in Manhattan. And I worked under Dr. Ann Geller, who was a neurologist. And we identified a protocol of neuropsychological tests that captured the cognitive impairments people came into treatment with because of their severe alcohol use disorder. And then we'd retest them after 28 days. It was pretty remarkable how resilient the human brain is despite being bathed in alcohol for a considerable period of time. And so that was my first experience with alcohol use disorder and with clinical research. So I know Ann a lot. And then I kind of set it aside and I did my dissertation research in the area of depression at Cornell University Medical College. And it was a huge center of depression research at the time. And that made it clear to me that a nice young clinical psychologist like myself would never have the opportunity to be a principal investigator because all the male MDs would fight over any depressed patient that came through the door. And I noticed that one of the reasons people were excluded from participating in the depression clinical trials was because they drank too much. And so I put in my first application to NAAA to study depression and alcoholism, which that's what we called it at the time. And I included all patients with alcohol use disorder. And about half of them also had major depressive disorder. And it was a double-blind placebo-controlled trial. And so half the patients approximately got dizipramine, which was the antidepressant of the day, and or matched placebo. And the results showed that not only did people with both alcohol use disorder and major depression respond just as well to antidepressant medication as patients without alcohol use disorder, but that if they weren't treated by placebo instead, they were more likely to relapse to drinking, and their depression remained unchanged from baseline after 6 months. So that piece was important because at the time, people felt, well, aren't all alcoholics depressed? And if you dry them out, doesn't the depression just go away? And so the study showed that no, the depression didn't just go away. In fact, it wasn't benign neglect because people were more likely to relapse to drinking if their depression wasn't treated. And in the group of people with alcohol use disorder who did not have depression, the antidepressant medication did absolutely nothing in terms of their drinking. And that's a finding that's been replicated over and over again with just about any antidepressant we can think of, such that the American Psychiatric Association, in their practice guidelines for the pharmacological treatment of alcohol use disorder, states that antidepressants are not indicated for the treatment of alcohol use disorder, which is, putting a fine point on that. So,
Yeah.
I want to highlight something You said at the very beginning about the 28 days. If I imagine myself as a patient, that sounds like a reason to be optimistic that there would be a lot of recovery in a relatively short time. But now we'll go back to your path.
Okay.
What challenges have you faced along your path?
Hmm. Well, I had mentioned the difficulty of being a young female Ph.D. and a very male dominant traditional uh, medical school setting. Uh, but I kind of rolled with punches and, you know, obviously it wasn't personal. And I actually learned how to write my first grant by reading a grant that um, one of the guys I worked with gave me to, I don't know, I had the references on or something. And that's how I learned how grants were organized. And I wrote my grant totally based on the organization of his grant. I didn't even know there were instructions for writing grants. So then I collaborated with Mary Jean Krieg, who was at Rockefeller, which was right next door to us, and did the aminopyrine breath test on my subjects who were being treated with disipramine. And at the time, people tended to give a little less medication to people with alcohol use disorder, maybe out of concern of drug interactions, or that, you know, they might take a whole handful of medication. So what the aminopyrine breath test showed was that, in fact, people with alcohol use disorder tended to have accelerated drug metabolism because of the alcohol use. And so they were actually being quite undertreated in terms of the amount of drugs circulating. And so that was also taken in and absorbed by um, the American Psychiatric Association. They highlighted that finding that I presented at their annual meeting. So, and then the American Medical Association had a press briefing of the results of the study, which was the cover article for JAMA. And so I just thought everybody who had a grant received in this manner, like, this is a new finding, so let's bring it forward. Well, I think that was the last time I was ever treated quite so receptively. um,
I know that a press release can be very exciting, a chance to get word out to the public. So, congratulations for that one.
Thank you.
What brought you to Scripps?
I had a stop in the middle. I wanted to go somewhere that really didn't have an established research infrastructure to the degree that Cornell did. And every year I'd go to what's called the NCU meeting, New Clinical Drug Evaluation Unit, Key Biscayne, Florida. And it was always around Memorial Day after, you know, a miserable winter and spring in Manhattan. And I said to myself one year, "You're a smart girl. Why aren't you staying here and going back to New York for meetings?" And so I just called up the chair of psychiatry and asked if I could transfer my grants to his department. And so I did. And I had a lot of fun there. Um, and had a great group of residents that I worked with. And um, the first thing I did was to acquire uh, nalmefene because it was being developed for interstitial cystitis by a local drug company, and a guy I knew from Rockefeller. I just moved there as scientific director, and he helped facilitate drug supply for a pilot study. And the basis of that work wrote an R1 to do, adequately powered dose ranging study, actually. And it was positive, and that actually attracted attention as well. It was presented to Congress by Dr. Varmus, who was the director of NIH at the time, as a pivotal study of 1999. And now, as you may know, nalmefene's approved throughout the European Union under the brand name of Celincro. And then also, while I was in Miami, I was asked to be the overall principal investigator of the acamprosate study, and served as consultant to the NDA, and was involved in every aspect of the FDA process, from pre-IND meetings to presenting the clinical dossier to their FDA advisory committee. And that was a good learning experience in terms of, how drugs move through the FDA approval process.
How long did that process take for acamprosate?
The FDA, or to do the clinical trial, or
The FDA, from when you started the process through getting it prescribed for people?
yeah, really wasn't that long, because it had a considerable clinical dossier from Europe. The US study was what it was. And so it was just a matter of kind of pulling it together and, making a plausible case for the endpoints and safety. And
May I back up a little to the previous drug?
yeah.
How did you make what sounds like a jump to a treatment for
Depressed alcohol.
cystitis?
Oh,
A leap from something so different.
okay. Because nalmefene is an opioid antagonist. And at the time, naltrexone had just gotten approved for the treatment of alcohol dependence. And nalmefene was a more potent opioid antagonist, and was also an antagonist, not just for the mu receptor like naltrexone, but also for delta, and more importantly for kappa, because kappa is the opposite of the mu receptor. And by blocking mu and kappa, the thinking was that it would function as a partial agonist, in that it would block some of the rewarding effects of alcohol, and at the same time block some of the misery of not drinking by blocking the kappa receptor.
Have you experienced at all what you might call a eureka moment? And how did you arrive at that point?
So I guess getting access to nalmefene was pretty much of a eureka moment. Being asked to do the acamprosate clinical trial, because I really like the creative part. I don't like to study the same drug over and over again, just like moving the needle a little bit. I like to get in and get dirty working with an entirely new compound, or new to our population, or, that has some element of freshness and creativity to it. And I've carried that forward at Scripps. And Floyd Bloom was the person who did my recruitment to his department. I think it was called neuropharmacology at the time. And Ernie Boyler, who was another chair of a department that was more clinically oriented, gave Floyd an assist. And Floyd's thinking was to have a fully translational department. You know, Floyd is one of the co-founders of Alkermes, which makes the long-acting naltrexone Vivitrol. And so he was very interested in having, you know, a fully translational department. And so I was his last recruit. And it was very exciting to me to be in that environment. I mean, Floyd, so awesome. I mean, he can just give the best comments. And of course, I was thrilled to work with George. And in fact, George Coop. So in fact, we had a grant together when I was in Miami and he was at Scripps. So that was part of the motivation to bring it together and align with Floyd's interest. And what that was to create parallel animal and human laboratory models of risk factors for relapse in people dependent on alcohol following that brief period of abstinence when you're vulnerable to hyperactivia. So, yeah, because George and Floyd's Alcohol Center were identifying so many neuropeptides that become overexpressed during that early phase and drive those clinical symptoms of anxiety and dysphoria, irritability and sleep disturbance, which then amplify craving and then, you know, you're right back where you began. And, you know, it was appealing to me because I had started out in depression and totally appreciated how this was a totally different affective phenomenon than major depression, you know, so I could get right into it. And I also like the concept of using a medication that could restore homeostasis to these brain systems that had become overactive in response to the stress of abstinence, and thought that could possibly take patients further than the older drugs like disulfiram, which made you sick if you drank, and naltrexone, which blocked the reward system. You know, and so and there were compliance problems with both of those medications, and I thought a drug in the kind of like the anti-stress domain would be more acceptable to patients because it wouldn't be involved with making them feel, you know, worse or bad. So that whole piece came together, and those were fun times. George recruited the most talented postdocs, you know, who would actually run the animal work. I still collaborate with them, and yeah, so that's been great to have people that I've worked with for more than 20 years, you know, where we really know where each other's thinking's coming from, and nobody gets stale. Like Olivier's gone from being Olivier George from being a lab manager to being, you know, a master of functional connectomics. So it's
The operation of your group sounds like you are positioned to take advantage of repurposing existing drugs to treat alcohol misuse. It looks to me like to be able to do that, you've set your work to span from preclinical to clinical work, a very translational operation. It's a very logical progression, fresh looks at available approaches. And did I did I get that right?
Got it right. Yes. Sometimes they repurpose. Sometimes we, are able to access an investigational drug. For example, I worked with mifepristone, which is a mixed progesterone and glucocorticoid antagonist that is, in the version that I used, is used chronically in Cushing's patients. And so I used the human laboratory model, and, the animal work was done. And, we have this iterative model where the human work informs the animal, and when you get things that correspond, results that correspond, then it's like thumbs up for the larger, longer, more expensive, adequately powered clinical trial, which we also did with mifepristone. But because of the controversy around its progesterone antagonist activity, I was really interested in studying a pure glucocorticoid antagonist, because Leandro Vengesculo, who's another cherished collaborator, who now has his own lab, his on tenure track at NIDA, he showed that it is the glucocorticoid antagonist activity of the molecule that has the alcohol effects. The progesterone has absolutely no contribution. So I think because I had behaved myself with the mifepristone studies, the drug was given to me by Corcept Therapeutics, that manufactures and markets Korlym, which is the version that's used for Cushing's patients. And they have a number of drugs in development. So they trusted me with one of their investigational drugs that is a pure glucocorticoid antagonist. So sometimes we get, there's no problem doing that work preclinically, but there are lots of risks to the drug when it's given to humans. So that was important, and it also confirmed the role of glucocorticoid antagonists for people in that early recovery phase.
There's the three medications that you've studied, described, found efficacy. Do you think that those will eventually end up available for patients for alcohol misuse in the US?
I think a glucocorticoid antagonist could. I think the company is quite committed. Um, it's not that any of the drugs couldn't make it through. It's whether there's a company willing to spend $10 million to, do the 2 6-month multi-center positive clinical trials. And
well, they have to make money.
yeah.
Some of this you've already discussed. What do you see as historical moments in medication development to treat alcohol misuse?
Oh, without a doubt, it was the finding by the Penn group. Joe Pacelli had the lead there, that naltrexone could reduce alcohol consumption and people who were dependent on alcohol. And then Stephanie O'Malley and her team at Yale completely replicated Joe's work and added on a piece that I also thought was valuable, which was as a 2x2 design, the naltrexone versus placebo, but also I believe it was cognitive behavioral versus supportive therapy. And it was the people who got the naltrexone plus the cognitive behavioral therapy that builds in all sorts of skills like drink refusal and riding the waves. That's a San Diego metaphor of great. So that was, to me, a total eye opener, because when I was at Cornell, the only treatments for alcohol were Alcoholics Anonymous, and we had meetings in the basement, like, at least once a week, because the self-help group, wrote every day of the week had a self-help group. They weren't always low alcohol. And disulfiram was approved in 1951. So those were the two treatments for alcohol use disorder. And disulfiram would have to be prescribed by a doctor who even knew it existed. And people would have to be willing to take it consistently. And I'm not knocking it, because I actually know people who are just like you and me who actually take it because they really need to be fearful of having a drink. So, but the emergence of naltrexone and in the US, and cambrosate was being developed in Europe, and then it coming together, all of a sudden we had options, So I think that really opened the door to a new world of possibilities for people with alcohol use disorder.
How have you seen the process of medication development evolve?
Well, one way we're very fortunate in terms of drug approval is that alcohol is actually detectable. I mean, I feel so badly for depression, schizophrenia, and all these psychiatric indications where all self-report. That's a tough thing to sell. And, we have self-report plus, alcohol glucuronide urine dipsticks can be sensitive to the last drink to 80-hour interval. Less if it was one drink, but if a person has had their usual walloping dose, it would be about that period of detection. So, and PEth is apparently becoming available in commercial labs. That's the alcohol glucuronide is actually, testing an element of alcohol. It's no inference. And PEth is one of those markers. And it's the most sensitive and specific, as I understand it. And it used to just be available, in an academic lab. But as I understand it now, it is becoming available in commercial labs, which is really great.
What challenges does the field face in medication uptake, and what can we researchers do to offset those challenges?
Well, we do have a very odd challenge. We almost have a civil war between the cambrosate and naltrexone versus the naltrexone proponents. And it's like in any other a specialty in psychiatry. New antidepressant, new antipsychotic. Yay, we've been waiting for this, And I just don't understand, kind of maybe understand the hostility with which a candidate was met because there was an established group around naltrexone, and it was an already FDA approved medication. It was approved in 1984 for opiate dependence. So there wasn't a problem. People could acquire it, for research purposes. But acamprosate was a new molecular entity in the US. And I think any company would not let a group of investigators with no relationship at all to the company have access to a drug. The company's paying many millions of dollars to move through the FDA approval process. So I think there was some narcissistic injury to being denied access to a acamprosate until it was approved. And people will say, well, you have to take it 3 times a day. And it's like, well, that's true for gabapentin too. And that's always one of the 20 most prescribed medications around the world. That dosing schedule didn't seem to interfere with its utilization. So I don't know, that's an odd thing. I think we'd have better implementation if that civil war could be resolved.
Sometimes things seem so strange and counterproductive. My grandfather had a quote. He told me that a man said to his wife, "Often I think that everybody's a little strange except me and thee. And sometimes I wonder about thee."
There you go.
For researchers at the start of their career, do you have any advice to offer? How about for basic scientists who want to pursue medication development and testing?
I do. So, Dusab Choi at the Mayo Clinic and his team did a pharmacometabolomic study of acamprosate and identified high baseline serum glutamate levels as being predictive of individuals highly likely to respond to acamprosate. And like a back check on it was the patients who responded to acamprosate follow-up serum glutamate levels had decreased with acamprosate treatment. So it's really solid. And I think for a young person, like this is an approved drug, there's no trouble getting it. It's like we really need to have that kind of information, those kind of markers that can identify, who's likely to have a good response on the first try, because you can go through one drug after another. We have 3 approved, and we also have 2, gabapentin and topiramate, that have been included in the American Psychiatric Association's practice guideline for the pharmacological treatment of alcohol use disorder on the basis of the work that independent investigators have done. And so now they can be prescribed in the VA system, and many insurance companies will cover it. I mean, gabapentin is so inexpensive, but it costs like maybe a few dollars for a week's supply or something.
You would encourage us to solve practical problems.
And collaborate with people who are doing clinical trials, which is a lot to mount until you've had experience, or even human laboratory studies, and bring something to it. develop an idea, interest, and bring it to the principal investigator and say, I'd like to contribute this. And then you have your data, your paper, and pilot data for your grant. And don't just stop at people in your own institution. I mean, I collaborate with people all over the world, because they have unique expertise in an area. They can do assays that I can't. And, I've had blood, breath, and urine travel all over. So there are a lot of possibilities.
For researchers in general, do you have anything that you would like to ask of us, of the research community at large?
Well, I really liked Vivek Murthy's Surgeon General's report on alcohol, drugs, and health, because he framed alcohol use disorder as a chronic disease like diabetes, where treatment involves both behavioral and pharmacological elements. The chart is tagged, at baseline, and the different elements of a person's life that have been affected by pathological drinking, and as well, of course, as the quantity and frequency of their drinking, so that at every subsequent visit, that will pop up. The diagnosis will pop up, so you can inquire about whether they're having any side effect to the medication, or in fact taking the medication. Do they have any concerns about their progress relapsing? And how has life changed, since they began making this effort? And I like to share the results of the liver function test panel that we get with subjects, and not just, shove it in the chart, but say, this is where you were when you came in, and this is where you are now. You're really doing something good for your health with the effort you're making. It's really showing. And I find that to be quite reinforcing.
For members of the public who may watch this video, do you have anything that you would like to communicate?
If you or someone you care about has a disorder, it can be quite mystifying about how to proceed. And I really like the general public to be aware that they just need to Google NIAAA, the National Institute of Alcohol Abuse and Alcoholism, and go to the treatment navigator page on their website, and that explains what alcohol use disorder is, the different evidence-based behavioral treatment options and medications available. And you can enter your zip code or your loved one's zip code and find treatment providers in your geographic area who practice evidence-based treatment for alcohol use disorder. And I think for the general public, just having that reassurance of how to proceed, that guidance, would be very helpful. And another thing that the general public and the research community both can help with is, alcohol is responsible for more deaths, just sticking with the CDC's use of alcohol being specified as a cause of death on death certificates. And that's an underestimate, but even using that, there are more deaths from alcohol than even from opioids. And I think tobacco is the only nicotine, the only substance that has more deaths than alcohol. And it's a tremendous burden on our economy in terms of the cost of treatment, lost work days, and court costs. I mean, it just filters in, to so many aspects of society in terms of costs and heartache, that you really want people to be as informed as possible. But on the other hand, you need resources, and NIAAA has a very modest budget, compared to other institutes, and it would be very worthwhile to contact your congressman and the people who represent your interests, to make sure that NIAAA has the share of the NIH budget or special appropriations that they really need, because it is a big problem, a costly problem, and potentially a lethal problem. And it's just phenomenal what NIAAA does and what its grantees do, with a limited budget. But if people could, make their representatives aware of how great the need is. Like, I looked up the where the medical examiner annual report of deaths caused by alcohol. It's causes, all different causes, but I looked for alcohol. And San Diego, there were 7 deaths due to alcohol intoxication, and we're a big university town. And so I just, surmise that at least some of those 7 were probably college students. Just heartbreaking.
It sounds like one message to convey to the public is that there is reason for optimism.
Yes. Mhm.
What do you envision for alcohol treatment in the near future and the more distant future?
So I think there is cause for optimism. People like me like to identify new treatments. People like to identify people who are more or less likely to benefit from a treatment. And now we have emerging treatments that are non-pharmacological, non-invasive treatments, like TMS, transmagnetic stimulation, for example. And so I think there are many pathways open to us to optimize treatment response for many more individuals struggling with alcohol use disorder.